My name is Lisa Giacomo. I am Kiah's advisor, and I'm going to provide a very quick introduction to her thesis defense today. So Kiah was one of the OC5, which doesn't refer to the very popular teen drama show from the early 2000s about the high-class world of Newport Beach, but rather the first original 5 members of the lab, Kiah, Caitlin, Rob, Jason, and Malcolm. Kiah was exceptionally productive during her time at Stanford as a student, and for each of her primary first author papers, I just wanted to talk a little bit about some of her innate talent and the interests that she brought to the table, and just highlight those before she presents them today. So, Kiah's first paper in the lab emerged from her rotation project, and I think she knew there was real promise early on, as evidenced by this message I found in 2013. It definitely looks like a real result. And in this paper, Kiah considered how environmental walls correct error in position codes, and I think this early success was inevitable given that I later learned she had extensive experience writing equations on environmental walls and boundaries as an undergraduate. Kiah's second paper leveraged her strengths as an accomplished athlete. For example, while a graduate student, she actually placed first in the highly competitive Silicon Valley Turkey Trot. These athletic skills came into play with her second project, which required her to become a very good swimmer. On many occasions, while working on the project, Kiah and I would have a long discussion about the analyses, and during these meetings, Kiah would tell me that she felt, in terms of the project, as if she was swimming in different directions in a lake that had no shore. Fortunately, she found the shore, and she created a really wonderful bridge between both theory and experimental work and published a very excellent paper. For Kiah's third paper, she leveraged her love of good food, and together with Will Butler in the lab, designed a task in which an animal had to navigate to a remembered reward location, and when it heard a tone, in order to receive a food reward, which is then rapidly consumed. And finally, Kiah wrote a really fantastic review article on heterogeneous coding and mixed selectivity, and I think this really leveraged how mixed her talents can be. She's excellent at camouflage. I doubt any of you can even find her in this picture, but she's actually here under all that grass. She's a talented hula hooper, and I think personally she shows real promise in sledding while wearing snowshoes simultaneously. In summary, Kiah succeeded because she's an excellent scientist. She shows mastery in computational and experimental realms, exceptional creativity in her ideas about how to advance and understand the brain. She has a keen intuition and raw talent for digging deep into a question and coming out with a fundamentally new understanding of the topic. She's also a pillar of our neuroscience community. She has taught math tools for many, many years, as well as TAing courses for the neuroscience program. And we can't wait to see where she goes next. So without further ado, Kiah.
For that lovely introduction. Also, thank you to all of you for coming today. I really, really appreciate it. I'm really excited to share with you what I've been working on for the last 6 years, which is investigating the internal code for spatial navigation. So navigation is something that we do every day. Perhaps you can relate to this because you just had to navigate here to the Clark Center auditorium. And to further demonstrate the kind of behavior that I'm interested in, I want to take this example just a little bit further, and let's pretend that you started at the Hoover Tower. So to get to the auditorium, you left the Hoover Tower, turned left on Sarah Street, and walked along, meandered through some buildings to get to the auditorium. You listened to my talk, and then you decided afterwards that you're hungry, so you're going to stop by Bites to get a sandwich, and then maybe take the scenic route back to the Memorial Court to where you started from. And maybe you've never even taken this exact trajectory before, but you can do it because you're familiar with this campus, and you've built up this map of what the campus looks like, and you can use that map to get back to where you started from. And I think it's very easy to take for granted the fact that our brains can do that, so we can build internal maps of the outside world and use them to accurately navigate. When the underlying computations are, I think, quite substantial. And understanding how the brain does this is also quite difficult. But that's exactly what I want to do. I want to understand how neurons in our brain can perform the computations necessary to allow us to accurately navigate. So before I dive into the neuroscience, I want to first consider navigation. How do we know where we are? How does our brain know where we are? And there are at least two different ways that we can figure this out. One is through landmark-based navigation. And this is quite simple. You just estimate your current location based on the perceived distance to a landmark. So if you can see the Hoover Tower, then you know you must be near the Hoover Tower. And this is good because it's accurate, but bad in that you need a familiar landmark. Another method is path integration. And this is where you estimate your current position based on a previous estimated position and the velocity in between. So the idea is just that you are adding up velocity over time to get a running estimate of your position over time. And this might be what you could use if you know you have to walk about 3/4 of a mile down Sarah Street, so you can just use your self-motion cues to figure out roughly how fast you're going, and then you know how long it will take to walk 3/4 of a mile. And this is good because you don't need prominent familiar landmarks in this case, but it's bad in that error can accumulate. Our sense of how fast we're going isn't 100% accurate. So as neuroscientists, the question then is, well, how does our brain encode these variables that we think are important? How does our brain encode things like distance to a landmark or position or how fast we're running or which direction we're facing? So there are many different ways that one could go about finding the neural basis of navigation. And in my PhD, when I joined Lisa Giacomo's lab, I decided on one particular route, which is to use model organisms like mice and rats. And we watch them navigate, and we think, okay, well, what are the individual neurons doing when an animal navigates? And specifically, how does the neuron activity correspond to these variables of interest, like distance to landmarks or position or head direction or running speed? So to do this, to answer this question, we will chronically implant tetrodes into the brain, and tetrodes are just a bundle of 4 wires. And if you snuggle up next to a neuron, you can then record a single neuron's activity, like this one in red. And if we hook this up to a computer, we can then visualize the neuron's activity over time. And neurons are active by firing spikes, which I'm denoting here by these lines. We will typically talk about neural activity in units of spikes per second, which I will refer to as a firing rate. So after we implant these tetrodes into the brain, we then want to watch an animal navigate. And the simplest task that we use is called a random foraging task. And this is simply when you just place an animal in an open, square environment, and you allow it to randomly forage for scattered reward. And we will typically just sprinkle Cheerios across the environment and have the animal run around and eat them up. And we have a video camera system set up that allows us to monitor the animal's behavior. And so now we can record spikes from a neuron, along with the animal's position, head direction, and running speed. And we can record these variables over, say, a 30-minute recording session. And now the question is, how does neural activity correspond to animal behavior as captured by these 3 variables? And this, at the core of it all, is the question of my PhD. But first, we need to address one thing, which is where to put our tetrodes. Where should we record from? And there are many places that one could record from, and I think find very interesting things in regards to navigation. But there's certainly a hub of navigational activity in the brain, and that hub is here. And there are many different regions in this area, but one prominent one is the hippocampus, which we've known for over 50 years is absolutely critical for spatial memory. If you take it out, then animals can't navigate back to remembered locations. But I didn't study the hippocampus directly. I studied another region called the medial entorhinal cortex. And I will refer to this sometimes as MEC or just entorhinal cortex. And MEC sends projections to the hippocampus. It's the major cortical region to send input to the hippocampus. And also, entorhinal cortex receives a lot of sensory information, like olfactory and visual inputs. And so it acts as this relay station between the rest of the brain and the hippocampus, therefore putting in a very good position to do important computations supporting navigation. I think also, though, the most convincing reason one should study entorhinal cortex is because what you find there is super interesting. And this video shows, I think, one of the most fascinating findings for the navigation field. And this video will also demonstrate the kind of experiments that we do. So this is an animal navigating in this open square environment. This is a rat. And you can see the cable here. This is connected to the wires that are implanted in the brain and are carrying a signal to the computer. And you can see that there is an experimenter here on the side. They're tossing in Cheerios to motivate the animal to run around. And there are these white diamonds being plotted. So a diamond is plotted every time the neuron that we're recording from fires a spike. And perhaps you can appreciate that the cell is not firing just randomly across the environment. It's firing in very specific locations. And what's really incredible is that these locations form a pattern. This pattern has a hexagonal symmetry to it. And we call this pattern a grid pattern. And we call the cell that we're recording from a grid cell. One way that we visualize grid cell data is through a plot like this here. This is a bird's eye view of the environment, and the black line is just the animal's path over the entire recording session. And the red dots are the locations of the animal when the cell fired a spike. We also visualize the data through a heat map, and I'll be using these frequently throughout the talk. This is the same cell, the same data, just shown a different way. And the color, every pixel in this map corresponds to the average firing rate of the neuron. So again, perhaps you can appreciate the pattern that arises, this grid pattern. And because of its such strict and beautiful geometry, people immediately concluded that this cell must form a metric map of position, perhaps somewhat akin to the latitude-longitude maps that we use. Grid cells, though, are not the only cell type found in entorhinal cortex. There are also border cells, and these are cells that fire maximally along one boundary in an environment. There are head direction cells. So here I'm plotting the neural activity versus the animal's direction. And this cell fires maximally at pi radians, which in this case corresponds to the animal facing west. And so because these cells are active in a particular direction, they've been likened to a sort of neural compass. And finally, there are speed cells. These cells will linearly increase their activity with the running speed of the animal. And because they're active at fast speeds, they have been likened to a neural speedometer. And these cells have been found across a number of different animal species, one of which you may recognize, which points to the fact that they probably serve as an evolutionarily conserved mechanism for supporting navigation. So I will paint for you now the classic picture of entorhinal function when I started my PhD. And that's that there are these very specific dedicated cell types. There are grid cells, border cells, head direction cells, and speed cells. And these cells very much functioned like the maps that we use, and that they don't change with our behavior or reward. A map is the same of an environment no matter how you feel about it. And the grand hypothesis is that these cells interact to perform path integration. So this is specifically, grid cells form the position estimate. The head direction and speed cells form the velocity signal. And these two cell types will send inputs to the grid cell network, which then is the thing that is integrating velocity to get an estimate of position. So during my PhD, I first wondered how border cells, which were the other cell type, could be brought into the mix, maybe as some kind of landmark influence. I then started to question if this was actually the entire picture, if this was the only way the entorhinal cortex supports navigation, or if there's something beyond this. And then finally, I started to wonder if maybe this system was a bit more flexible than we thought. There's actually no reason the brain has to act like the maps that we use. So these questions came through in 4 different stories that I'll tell you today. The first is regarding the landmarks and how they can influence grid cell path integration. So the general idea here is just that maybe you're navigating back to Hoover Tower. Maybe you're path integrating back to Hoover Tower. And you start to get a little bit off because path integration isn't 100% accurate. But when you see the Hoover Tower, you know where you are because you can do landmark-based navigation now, and maybe then update your internal representation accordingly. And they wanted to know if this was reflected in the neural system. I then investigated whether cells could encode navigational information in other ways. And then I investigated how internal maps could potentially change with behavior. And I focused on running speed because of this one behavior that is changing all the time when we're navigating. And then finally, I looked at how internal maps could change with reward because reward is so important in determining where we actually navigate. Okay, so starting with the first story. As I told you, integration of a noisy velocity can result in an accumulation of error in our location estimate. So even if you start with a very accurate estimate, if you're adding up numbers that are just a little bit off, you will, your estimate of your location will diverge from the actual location. But if you see the Hoover Tower, then you say, right, I'm near the Hoover Tower. I know where I am. I should update my neural representation accordingly. So maybe error can be corrected through landmarks. But we don't have animals navigating to the Hoover Tower in the lab. We have them in this open environment. And this is in a dimly lit room, so there aren't a lot of sensory cues in the center of it. But they have this border, which is a very salient landmark. And so the idea is that maybe if an animal runs into the boundary, then they can say, oh, right, I'm at this wall. I now kind of know where I am. So if grid cells are the thing that's path integrating, then we predict that errors should accumulate when the animal is far from the border. And that error should then be corrected by the border. And if these things are true, then we would like to investigate a neural circuit mechanism by which this error correction could occur. So first, to investigate error accumulation, we looked at a set of grid cells. These were cells actually recorded by Lisa herself. So these were cells that were recorded in the environment during the random foraging task that I told you about earlier. And I first identified the firing fields of all these grid cells. And I then identified a boundary region, which is highlighted in yellow here. So the idea is just that when the animal is in between this yellow line and the edge, they have enough sensory input to know that they're near the border. I then looked at trajectories, or times when the animal left the boundary and wandered around and then re-entered. And I looked at spikes along these trajectories. I computed the distance of every spike to the nearest center of mass, and I took this distance as my readout of error. So if a spike is really far away from the center of a firing field, that's a high error spike. I can then look at error as a function of time from the last border touch or distance from the last border touch. And you can see that in this one trajectory, error is increasing. Now the question is, what do you see across all trajectories? And we saw that on average, there was this increase with both time and distance traveled from the last border touch. So to verify that this was not due just to the fact that neurons are noisy, I did a number of statistical tests. And in one of them, I simulated an entirely new data set that was just like the actual data. But in a simulated data set, I only created spikes according to Poisson statistics. And so I effectively removed any potential error accumulation in this system. I can then run the same analyses and figure out what I would get if there was no error accumulation and generate these null distributions. And the data that I observe in this is in this red dotted line, which is much higher than what you would expect given the null hypothesis, therefore suggesting that error does increase with time and distance traveled from the last boundary encounter. So error is accumulating when the animal is far from boundaries. If borders are really correcting the error, then the error should actually be corrected in a very specific manner, which is in a direction-dependent manner. And the idea is this. Let's say that the animal is in the environment and there's some estimate of the location that's a little bit off. And we can compute how off it is in the north, south, and east, west components. Now, if the animal runs into, say, the north boundary, then the estimate should be updated along the north-south direction. But the animal doesn't have any information about where it is along this direction, so that error can't be corrected. And then when the animal leaves the environment, then the error should be smaller in the north-south direction compared to the east-west direction. So what I can do is look at these trajectories when the animal leaves from a particular boundary region and look at the spikes, in particular the north-south and east-west components of the error of these spikes. then I can ask things like, is the north-south error smaller after a north-south compared to an east-west border touch? And that's exactly what I found, that the north-south error was much smaller after a north-south compared to east-west border touch, and conversely, the east-west error was much smaller after the animal touched an east-west compared to a north-south boundary. So this tells us that error correction is related to the position information conferred by a landmark, which is then further support for border-driven error correction. So now that we have this evidence of error accumulation and boundary-specific error correction, we want to know if there's, or we can think of a network-level mechanism by which this could occur. So we turn to this classic picture where grid cells are integrating a velocity signal from head direction speed cells. And we wondered if border cells, which very conveniently encode the boundary of the environment, could somehow be participating and serve as a neural substrate for error correction. So we've developed a mechanism that I will schematize for you here, and this had also been brought up several times in the literature prior to this project. So we can model a grid cell network as neurons in a neural sheet. So every point here, every dot, is a neuron, and I just arranged them to be in this neural sheet. And some neurons are active, they're in red, and other neurons are inactive, they're in gray. So when the animal's in a particular position, right, there's some cells on and some cells off. All the cells that are on are cells that have a field when the animal's in this location. And the idea is that if the animal wanders around in the environment, then error will accumulate in this system, and that comes through in this picture as the wrong set of neurons being active. But if a border cell knows which neurons should be active, then it can drive the correct activity pattern, and therefore correct the error. And Sam Ocko, who's an extremely talented postdoc in Surya's lab, has really beautifully extended and reframed this whole problem to show how border cells can learn this activity pattern as opposed to just know it. So the high-level idea is just that when an animal is in a boundary, then the border cell will drive the correct activity pattern in the network. So I took a model that was previously published and extended it to include border cells. And now we can see what we get. After 10 minutes of this simulated data, there's no difference really in the grid pattern in a model with border cells or without border cells. But as time goes on, a model without border cells will start to fall apart because there's no way to correct this error. But if you have corrective border input, then the error can be corrected, and the grid pattern will be maintained throughout the entire recording session. And I want to note that we can record grid cells for 30 minutes. So this is a much better picture of grid cell activity than this one. And then I was able to go back and reproduce all the experimental results that I showed you with this computational model. So in conclusion for this first story, we found that landmarks do appear to correct accumulated grid cell error, and we developed a mechanism by which this could happen. So next, I started to wonder whether cells could encode navigational information in ways other than what we've been talking about so far. So there's this very specific picture that there's a neural map and a neural compass and a neural speedometer, and more even, that every neuron just has one job. There's like a speedometer neuron and a compass neuron in your brain. And I just wondered if this was really the case or if there was just more to the story. So to paint the picture, like I said, there are these 4 cell types, and we identify every cell type using what I call the score method. And the score method is just a way to pick out the cells that will look like one of these types. So the grid score will characterize the 60-degree symmetry of a position tuning curve, and so a cell with a high grid score will be a grid cell. A border score computes the degree of the amount of activity along the boundary, and a head direction score will compute how much activity is in one particular direction, and so on. So they all compute a metric of interest about a tuning curve. And the classic pipeline is that you record cells, and you compute these tuning curves, you compute the scores from the tuning curves, and then you just see if that score is above some threshold. And this pipeline has led to the discovery of these cell types, which has been hugely important. It led to this hypothesis about path integration and hypotheses about how neurons in this region can be connected, and it's been absolutely critical. But there's a flip side to it, which is that the simplicity of these cell types, the fact that the grid pattern looks so beautiful and these head direction cells look so nice, means that people often will only study cells that look like this. And this would be fine if all cells could be placed into one of these categories, but that's really not the case. This is a pie chart that I made very early on in my PhD when I just collected data from a bunch of people in the lab, so I was just curious how things really broke down. And I saw that the majority of cells that people recorded actually didn't fit into one of these categories. And also, a very small number of cells were conjunctive, which means that they encoded multiple things. So a grid head direction cell is a conjunctive cell. So now the question is, well, what is going on in this section of the pie chart? And cells in this section will have kind of funky looking tuning curves. This is one example position tuning curve. This isn't a grid or border pattern, but it looks like it could still be informative about the animal's position. Here's an example head direction tuning curve. Again, it's not the classic encoding for one direction head direction cell. And here's a speed tuning curve that isn't fitting the classic linearly increasing with running speed, but still could be informative. So the question that I had was just, how can we classify these cells and understand their properties? It seemed like there was going to be no way to understand what entorhinal cortex was doing or how it was supporting navigation if we don't even know what most of the cells are encoding. So instead of asking whether or not a cell looked a certain way, like if it had a high or low grid score or a high or low head direction score, I instead asked a much more general question, which was, does the animal's position, head direction, or running speed predict cell spiking, and how does it do that? And this type of question fits very nicely into a statistical modeling framework. And for the aficionados in the audience, I chose a linear nonlinear Poisson model, which is basically just like fancy regression. This model will take information about the animal's position, head direction, and running speed. It will then learn a mapping that will tell us an estimate of the cell's firing rate over time. And we can compare this estimate with the actual firing rate. We want these two things to match as closely as possible. And in order to make them match, we will alter this set of parameters, which is the mapping. And these parameters will indicate how a cell encodes that variables. These are somewhat analogous to the tuning curves I've been showing you. But I wanted to learn one extra thing, which was, which variables does a cell care about? I want to know the minimum set of variables that are needed to predict spiking as accurately as possible. You can imagine maybe a cell only encodes position, then I don't want this position and running speed stuff in the model. So to do that, I just use a forward search model selection or feature selection procedure, where I just started with one variable at a time and then added things in until I stopped seeing improvements in my model fit. So I then returned to this data set that I looked at originally, and I just ran it through the modeling pipeline and compared it to what comes out of the score-based pipeline. And I was able to generate a new pie chart. And in this pie chart, I see that there's much fewer cells that are labeled as not identified. So meaning that all of the cells with the funky tuning curves, or a number of them at least, are encoding important or relevant information. And I also saw that there were a number of cells that encoded multiple variables. And so this is the area of the pie chart that has multiple variable coding. This is the corresponding area in the score-based pie chart. And this is quantified in this bar plot here. So this means that our picture of one neuron, one job isn't the complete story. Neurons are multiplexing signals in entorhinal cortex. This model is also able to uncover diverse coding properties. So I can now pull out cells that don't have a grid or border pattern, but are still informative about position. And same thing for head direction cells. This doesn't have the classic unimodal tuning, but it's still informative, and I can now look at it. And the same thing with speed cells. And I saw really there's just a whole large degree of diversity in how a cell can encode information. Like, the tuning curves are just extremely heterogeneous. So in conclusion, I saw that cells were not just coding one variable at a time. In fact, they were mixing variables. And cells can encode information in really diverse ways. It goes far beyond this classic 4-cell type picture. So now, given that I saw that maybe there's more to entorhinal cortex than this grid cell path integration story, I started to wonder if entorhinal cortex could also change with the animal's behavior. There's a lot other stuff going on, maybe this could be going on too. And I wondered if entorhinal cortex was acting like the maps that we use, or the compasses or speedometers, or if it could be something different. And I wanted to focus on running speed just because this changes a lot. And the picture in my head is just that, maybe if you're like me and you're late to every single thing, except for I wasn't late to this. And you're like, we're running down Sarah Street, which is what would happen to me if I wasn't have to be here. And you're focusing and you're going really fast. Is anything different in entorhinal cortex in that case? There's also a neuroscientific reason to look at this, which is that sensory neurons are known to adaptively change their coding properties when the statistics of the sensory input changes. And we know that sensory cortex would also reflect changes in sensory input. So to investigate this, I took every session and I split it into times of fast and slow running speed. And then I fit the model to each section separately. So I have a model that's just looking at position and head direction. And then I can look at the model output. So this is an example for one cell. And I'm plotting model performance on the y-axis and the different kinds of models on the x-axis. So the black points here correspond to the output when the animal is running slowly. And in this case, the cell is just encoding head direction. There's nothing informative about position. But when the animal's running fast, which is in purple, the cell encodes both position and head direction. And across the board, the performance is much higher, which means that I can better predict the cell spiking using the animal's position and head direction. We saw this for a number of cells in the data set. They tended to, more of them tended to encode position and head direction. Many more cells exhibited conjunctive coding as well. And interestingly, the unclassified cells were much more likely to gain features with running speed than the typical classified cells. I can then look at the individual tuning curves and see how they're changing. And I saw that when the animal was running fast, tuning curves were much more informative. So this is the same cell, just when the animal's running fast and when it's running slow. And this is the head direction tuning, again, for a cell when the animal's running fast and slow. And in both cases, I hope you can appreciate that the amplitude is much larger, which means that this is much more informative for the brain. So at a high level, this tells us that entorhinal neurons are dynamically changing their tuning properties with the animal's behavior. It's not this fixed map. So in conclusion, entorhinal maps of space can change with running speed, and in particular, they get more informative at fast running speeds. And finally, I wanted to look at reward. And I was encouraged to look at this because I saw that things were a bit more flexible than we thought. And reward is really important to us in determining where we navigate. And I was just curious, if I go to Bites all the time for lunch, is this location now special to me, or is it just like any other location on campus? Also, I want to point out that in all the experience we've done so far, it's just an animal randomly foraging for reward in this environment. There's nothing special about any area. And so we wouldn't be able to pick this out so far in the experience we've been doing. But this contrasts with a lot of ways that we navigate, which is typically towards a remembered goal location. So given that cells can be flexible with respect to behaviors like running speed, could they also be flexible with respect to reward? In order to do this, I turned to recording of rats, and we recorded from rats in two separate environments. One was a random foraging environment. This environment had black walls and was scented with lemon, and the animal just did the very classic random foraging task. In the second environment, it looked and smelled differently. There were white walls and was scented with vanilla, and the animal had to do a relatively simple task. There was an invisible reward zone, and when a tone came on, the animal was trained to navigate directly to the reward zone. When it did that, it got an extra large Cheerio reward, and the tone, big deal, and the tone would turn off. This is an example data from a single session. The gray line is the animal's trajectory over the entire recording session. This is where the reward zone would be, but it's not relevant in this task. I just want to show you for visual aid. And again, the gray line in this case is just when the animal is randomly foraging in between trials. And all of the colored lines are the times when the animal is doing this task. So in this case, the tone turned on here, and the animal navigated to the reward zone. So the question now, we recorded about 800 cells in 7 rats, and the question is, how do cells in co-position, or the cells that in co-position, how do they respond to the reward? And I would really like to highlight that all of this work was done with Will Butler, who is a really talented postdoc in Lisa's lab, and he just left for a fancy data science job in Minnesota. So we first looked at grid cells, and we wondered if their tuning curves changed in the rewarded context compared to the random foraging context. So we looked at their tuning, and we saw indeed there was a change in the grid patterns. These do not look identical. So we computed various ways in which the tuning curves change. I'll just share with you one today. And we looked at the orientation, which is just an axis of the grid pattern. We can compute the orientation in the random foraging environment applied against the orientation in the task environment. And we saw that often the points, which represent an individual cell, did not lie directly on this red line, which would be the case if it was the same, but they lied off the line. So this means that the map that your brain is using is rotating between the two environments. But we have a thing we need to settle, which is that this could just be due to the fact that we're recording in environments that look and smell differently. Maybe it has nothing to do with the reward at all. So we did a set of control experiments in which we had the animal do a random foraging task in both environments. And then we can do the same analysis and look at changes in orientation. And in this case, we didn't see any change at all. So this tells us that changing what an animal does in an environment will change the grid pattern in that environment. We then looked at how non-grid position cells could change their tuning. So I returned to the model from the second project and the set of these position cells that had funky-looking tuning curves but weren't grid or border cells. And I can then look at what they look like in each environment. And again, we see across these 2 cell examples that they also change their tuning. And 2/3 of the cells remapped, which just means that they significantly change their tuning curve across each environment. We have to ask the same question, is this due to differences in color or scent, or is it actually due to a difference in task demand? And we looked at the same control experiments. We can look at tuning curves in this case, and we saw that on average, the tuning curves were much more similar when the animal had to do the same task in both environments compared to when it had to do different tasks. And we can quantify this through correlation coefficients, which tell us just how similar these maps are, and correlation coefficients in this case were much smaller than in this case. So this tells us that, again, changing what an animal does in the environment will change the activity in non-grid position cells. So given that we see cells are remapping, we wondered if the remapping was reward specific, because that's the most important change to the animal. So to look at this, we first again looked at grid cells, and we considered the activity as a function of distance from the reward zone. And we asked if there was any change that was occurring as the animal got closer or farther away from the reward. So we can plot the average activity of all grid cells for the reward task environment as a function of distance, and same for the random foraging task. And when we compute the difference, we see that there's much more activity near the reward zone when the animal's in the reward task environment. So in this particular case, cells are firing more near the reward. We can do the same thing for the non-grid position cells, and I hope you can appreciate even this. In this single example, there's much more activity near the reward. We can again compute the activity as a function of distance. And again, we see that there is much more activity near the reward zone when the animal is in the reward task environment. So this means that both grid and non-grid position cells are changing their activity in a way that emphasizes the reward location. We then wanted to know if there is anything special that was happening when the animal was actually doing the task. And so if you recall, most of the time, or a pretty large chunk of the time, when the animal's in this environment, it's just randomly foraging. These are like the in-between trial times. And only every now and then is this tone coming on and the animal's doing this direct goal-directed navigation to the reward. I just wondered if anything special was happening during those times, or if really the most salient thing was just that there was a reward and the animal cared about that location. To investigate that, we took a full session data, and then we split it into times when the animal was doing the task and times when the animal was not doing the task. And this is just a single cell example. We can then compute the activity as a function of distance from the reward zone. And in this particular case, we see there's almost no difference when the animal is actively doing a task or when it's just randomly foraging. This cell just cares about this location, kind of no matter what the animal's doing with respect to goal-directed navigation. And we saw this across the board for both grid cells and for non-grid position cells. So they did tend to increase their activity, just like we saw in the previous slide, with distance or proximity to the reward zone, but there's no striking difference between task and no task times. So this means that the reward-related remapping that we saw is reflecting a long-term change across the session, and it is not just tied to the specific times when the animal is actively navigating to the reward. And finally, we investigated how these changes could impact navigation by decoding the animal's position in each environment. So here I'm showing the output of a decoder for a single session. These are now new kinds of rate maps, where the color of every pixel corresponds to the error of a decoder. And you can see in the reward task environment, there's quite low error, and in this case, there's actually high error near the reward in the random foraging environment. We can look at the difference between these two maps, and you can see quite clearly that there is much lower error comparatively in the reward task environment compared to the random foraging environment. And we can quantify this by looking at the error as a function of distance from the reward zone. And we see in this case that the decoder error is much smaller in the reward task when the animal's near the reward zone. We can now look across all of our recording sessions and see if this holds up in general, and it does. The decoder is much more accurate when the animal is near the reward zone, and this means that these changes in firing that we have observed are supporting improved decoding at that location. So in conclusion for this chapter, entorhinal maps of space do change with reward, and they change in a very specific way in which they increase activity near the reward. So finally, just to summarize, I showed you that landmarks can support accurate grid cell patterns, that entorhinal neurons encode navigational information in very mixed and diverse ways, so it's not like the maps that we use. Our brain does it a different way. And finally, I showed you that entorhinal coding is flexible with respect to animal behavior and reward. So I think the work that I showed you brings up a lot of future questions that I think are really interesting. One of the most fascinating questions is, how could this network be structured? How do you have things that look so clean and beautiful like grid cells nestled in a circuit with things that look so messy and flexible as all the other cell types? And how does everything work together to support path integration or navigation, generally speaking? I also wonder with what other variables entorhinal cortex could be flexible. And so perhaps this running speed analysis is really just driven from attention or arousal. And this is one project that people in the lab are currently looking at. And then finally, one thing I've always been kind of curious about is what entorhinal cortex does for navigation beyond path integration. I think that maybe this flexibility and diversity could be signatures of another computation that's going on. And with that, I would like to do a short acknowledgments before my personal acknowledgments. First, I would like to acknowledge Lisa, who steers the great ship of the GeoCoMo lab. And I would like to thank everyone in the lab for all of their feedback and support. Will, who did the reward project with me. I'd like to thank Surya for all the guidance and the clarity of thinking, and the Ganguly lab for all their input, and Niru, who helped me a lot in developing the model and honestly taught me everything that I know about statistical models. I'd like to thank my thesis committee and June Ding for chairing this thesis defense today, and the neuroscience program administrators, Ross, Katie, Kalai, Ernie, and Pardeep. A special shout out to Kalai, who is just a superwoman. And the neuroscience program directors, John Huguenard and Tony Ricci. I think this program is just beyond fantastic. and that's in large part due to the directors. And then finally, I'd like to thank my funding, the NBC for giving me money, and my current funding, the SIGF, and also the Stanford Neuroscience Training Grant. And with that, I'll take any questions.
Thanks. That was awesome, Kiah. I was wondering, in the last part of the talk, the task with two different environments, you have two cue cards, white and black. I'm wondering if there is like a rotational invariance in how the grids might be represented. Are the rats really treating the black and white cue card as the same object in the two different environments?
That's a great question. So I would say that's like a plausible hypothesis, but looking at the data, I don't think so. And that's just because of the way the rotations are. They're different across animals, for example. And the cue cards are in the exact same place for both environments. And yeah, so I,
okay. So when you do the comparisons, you sort of search over all four rotations?
Yeah, we never see rotations that are, I would say 90 degrees kind of drastic.
Okay.
Yeah, a lot of them are much more modest, like the 20 degree range. Yeah.
Thanks so much. That's awesome. On your fourth story, the reward, what do you happens when you're in an environment with many rewards, or rewards that change with time, so your model has to be updated?
Yes, such a good question. So there was a complementary story, a project done by another lab looking at reward in enteral cortex and also in hippocampus, and they had multiple rewards in that case. And they saw that their task was a bit different, and so their results were a little bit different as well. But they saw that something happened with every single reward location. And in their results, the fields were actually kind of being moved a bit closer. And so they saw that across the different rewards. So I would expect in this case that we would just see increased activity across any rewarded location. Yeah.
Thanks, that was really interesting. Do you have a sense from looking at your decoders why increased firing rate near the reward zone improves decoding performance?
Yeah, so I think there are multiple things going on. First is that there are more spikes, which can be just helpful to decoders if you have more information to play with. Enteral neurons are also very low firing rate, just at a baseline, and so having more of that will be helpful. But also, I think that there's another thing at play. So if I downsample the spike numbers to be the same, then I still see an improvement in the decoder. And so my pet hypothesis is that the system is paying more attention to the animal's position because it's very relevant when you're near the reward zone, and so the spikes are also closer tied to the position. Yeah. So my take home is that spike number is helping, but also what could be driving this effect is that the spikes are better tied to position. Yeah.
Yeah. In the last example, do you see any changes in the animal's behavior that suggest that it's learning more quickly, or that it has is more aroused in some way, and that is leading to some of the changes in behavior?
Yes, that is also possible. So to control for differences in running speed, which we did see, because the animal was booking it to the reward zone, we just downsampled the data to match in running speeds across the two environments as an attempt to control their behavior. We also were concerned, actually particularly about whether the fact that the animal spent so much time at the reward zone, they kind of hovered around it. Maybe that was enough to just drive these results. And so we did controls looking at other areas where the animal would spend a lot of time and we didn't see any change in activity. But higher level things like the animals paying more attention when they're in that location, I think we'll need a separate set of experiments to look at, but I think it's super interesting. Yeah.
So you nicely showed that the running speed makes the cells more kind of not ambiguous, but they're more fluent as to what they're encoding.
Yeah.
So my take on that, which I'm curious to hear your thoughts, is that it's less reliable, perhaps, because the animal's moving quicker, so there needs to be more compensation, perhaps, in the cells. Do you think in a more complex environment, beyond just like a border, cells would show more or less encoding of other information, or do you think they become more specialized because there's more information to use in the environment?
Yes, that is a super good question. So just to touch on your first part, the way that I think about it is that if you're running really fast, then your position's changing really quickly, and so you can't screw up. You have to encode it accurately, or else any downstream region could be confused. And so you maybe put more resources into doing it well. And so I think that maybe that's a very general picture. So if the animal, it was very important to know where you are or what direction you're facing, then maybe that would have the same effect. Maybe if you have also more information available to you to compute your position or head direction, that could also influence it. I will say though that interestingly in these experiments, there's not a lot of position or position cues or cues about the environment, and the system does pretty well. So I think it's actually a very interesting research question. Yeah.
How long does it take to learn the tasks of the animals? Is it a kind of thing where you can record through the process of learning and see how the reward changes the rotation direction?
Yes. That is such a good question. It is possible. Did we do it? No. So it only takes about 2 weeks. But for a number of reasons, we actually decided to train the animals before implanting them. Just then we know which animals are good ones, stuff like that. And so we were then not able to look at things like learning. But I think it's a super interesting question to see kind of in real time how the system is changing to disambiguate these two environments. And there's been other work to look at it. This other paper that I mentioned earlier can also look at that kind of thing, but we do not. Is that it? Okay.
Kiah, before you start, this
boy.
is just from the Ganguly lab. Graduation Oh my gosh. This is really good. Okay, I'm just going to put this to the side. Okay. Some funny things on top. So I have some personal acknowledgements. First, I would like to thank the Queen, Lisa, for everything that's happened in my PhD. I think joining your lab was the best thing I could have ever done, and I'm so happy I did. You just made things flow downhill to success. And even in the hard times when I came to you and said I was swimming in a lake with no shore in sight, your response is not, Kiah, you're so melodramatic. But it was instead, let's have a meeting and let's just get a result for you that you can look at, and then maybe you'll feel a bit better. And I did. I felt a lot better after that. I appreciate so much all of our meetings and all the guidance and just the attitude that every problem has a solution. And you just lead so much by example with the enthusiasm you bring to work every day. And it's just, it has made everything fun. It's made everything so fun. And I was so sad a year ago when I realized I had to leave. Because I didn't want to. I think I'm just this could be the best thing that's ever happened. I don't want to leave. And also, just going back, Lisa has a side to her that we should all appreciate together, which is that she's a huge nerd. As evidenced
evidenced by the fact that she made us take this lab photo. Best lab photo ever. And she wrote this amazing quote in lab meeting the other day after the final Game of Thrones episode. And she said she's been thinking about who will be the Iron Throne as long as I've been thinking about the hippocampus. And she also told us, after she won our Game of Thrones competition thing, that she was once invited to be the key holder to the largest public library of sci-fi fantasy, which, it seems like a big deal. Also, to get more dirt on Lisa, I heavily stalked her husband, and there's like no email in sight. So I'm like locked out of my first LinkedIn account, so I had to get a second LinkedIn account to just like message him there. But because he's not in my network, I had to sign up for like premium. I sent him this like really weird message. They're like, "Hi, Ian, I'm defending, and so I'm just wondering if you have like any dirt on me that you can send me." And he sent this picture, which is like so innocent. So, you kind it well. I would also like to thank, from like the bottom of my heart, the Geocomal Lab. It has just been the best like environment I could ever imagine, and this lab is like a family, and it made even like the hardest days fun because it could be like a hard day in the company of people that I like. And I feel like I've learned so much. I learned so much about so many things, like learning about science and having my ideas challenged, and like hearing people talk about their own ideas has like brought so much growth in me. And also just like there's amazing resilience and passion and like hard work in this lab. It's the real deal, and I'm just so grateful that I was part of that. Caitlin, don't cry because I'm gonna cry too. I also though looked this up recently. This was like the very first Facebook message that I got, and it was just, I think it's just like really telling. Like Caitlin was the very first one, and she was like, "I am so excited, it's gonna be awesome," and it was really awesome. And I knew this lab was a family when I walked into lab. This was like right after I joined, and someone, I think it was Rob, took a screenshot of the background of my computer, like with the folders, and then made that screenshot my actual background, so I couldn't click on anything. And then he put like all of my he deleted all of my real folders and put it in the mystery folder, which is like hidden in the corner of my desktop. And so I came in and tried to click, and I was like, "What is going on?" And I actually still have this folder on my computer. And then also, Lisa, thank you for not kicking me out of your lab when I conspired to put a Christmas tree in it, decorated with Surya's face. I know you probably walked in and were like, we thought it was funny, so yeah, it's been such a good time. Surya, I would like to thank you so much for like everything you've done to guide me, and like every time I met with you, I felt like I grew a lot, and the ideas in my head that were like fuzzy and confusing would come out of your mouth as like, you know, like arranged and organized, and like everything makes sense now. Also, I'd like to thank you for giving us some gem quotes. Like, let's see if I can play this. So we had this video for my second paper, in which we, talked about the work and stuff. And Surya had a real gem at the end of it, which I think actually is pretty good, but it's really funny.
And we somehow need to break free of the prejudices of our mind in order to properly understand the brain.
It's a good one. Also, Surya made, actually made us put an end-final dark matter for the unclassified cells, which will live on in infamy. And also, oh, it's a little too dark, you can't see it, but this is your roasting. This is a photo of Surya singing Spice Girls karaoke, which Lisa has her nerd side. Surya has.
You don't have the video, right?
I do have the video, but I felt bad doing too embarrassing. But I have it and I watched it many times. I'd also like to thank the Ganguly Lab. I learned so much from you guys, and then every time I sat in lab meeting, even when it just washed over me completely, I feel like I learned a lot, and you helped me remember what math is, and yeah, and I would like to thank Niru in particular for just teaching me so much, and also guiding me through a lot of things I just didn't know how to do. And it's been a really fun time. I've enjoyed all of our conversations and talking about science and talking about my project, and you guys never shy away from asking the challenging, difficult questions, and it really helped me grow. I would also like to thank the neuroscience community. When I started this PhD, I was horrified. And over the 6 years, I just feel like I don't know, it's such a loving and supporting place full of just brilliant and inspirational people. And I grew to feel more comfortable and excited. And I think that there's something really special in this program. And I just am so grateful to have been a part of it. And there are a lot of fun times I really enjoyed, the retreats, and also thank you for letting me teach math tools to half the program. This is Lane and I conspiring on how we're going to grade the finals. And things like Super Friends and boot camp. Caitlin, I'm really sorry, you look so horrified in this photo. But they were just a lot of fun times, even hallway chats and just random happy hours. It's just been the best time. I would also like to thank, as far as I'm concerned, the most wonderful cohort to ever exist. I am just so happy to know all of you. Everyone is just incredible. I have so much respect, scientifically and personally, for the way people attack challenges and get through a PhD. And it's been a lot of fun going to the city and doing backpacking trips and doing dinners. And I just knew I always had a family that I could go to if there were hard times. And I am just endlessly grateful for them. In particular, I would like to thank the Stanford ladies for literally pulling me through sometimes. There have been a lot of random fun moments, like Caitlin saying she's totally here for teen romance. And, making these gemstone rock things and having dance parties in the woods and going on hikes and weddings and trips in New York. And it's just been there aren't words. There aren't words for how much love I have for all of you. And I'm so happy I get to live this life where I know you. I'd also like to thank Nick. Thank you so much for all the support and for believing in me when I didn't believe in myself, and for being there, just even if it was a good day or a bad day, and you saw all the moments of all my projects, and which are part of my soul in some way. And I can't thank you enough for helping me get through all of this. I'd also like to thank my sort of extended non-Stanford neuroscience friend group who have been critical to my journey. I am really grateful to have friends from college and high school and new friends at Stanford that I have met, kept in touch with, and it made everything fun. And it was a place I could go, people I could talk to when I needed a break from doing science. And also to shout out to the Stanford Running Club, which provided a place for me to, work hard in a very different way. And I think there are a lot of parallels between distance running and doing a PhD. And I realized this especially when Patty Sue, who was the coach for a long time, would tell me things that also seemed relevant to my entire life. There was one time when I was running a 5K race and she just yelled out, "Don't be a wimp!" And she's the grittiest person to ever exist. I was like, okay, I won't. And I think just having someone cheer for you when you're literally trying as hard as you can was just it's just such a nice metaphor for the rest of this journey. I also like to begin thanking my family that has come to watch me. So Shannon, thank you so much for coming here. You are the biggest badass to have ever lived. She is. And you are such a good role model for what it looks like to work hard and achieve your dreams. And I look up to that so much. And Jim, thank you for coming here. You make everything fun. And in particular, I think that you have showed me how to have joy in a challenge. And I think that's important and special, and I try, whenever I'm around you, to try to capture how a person can do that. I would like to thank my siblings, which, apart from being alive, is the best thing in the entire world to me. My brother, who never lets me off easy of any conversation I've ever had with him. And my sister, who lived with me for 3 years of the PhD and for 18 years before that, is a part of who I am. And she's my best friend, and I think it's very special when your best friend also has to be blood related to you and love you unconditionally. And you make everything better. You make everything better in this world. And even when it's ridiculous moments and you're talking about how saggy your pants are and mine's slim. Or having dance parties for Halloween and birthday carriage rides and going skydiving. And this is the cutest photo of us I think ever taken. You're my soul. And then finally, I would like to thank my parents. So whenever first I want to thank you for raising me to be a semi-decent human being that can do something in my life. But on top of that, I want to thank you for all of the gifts that I only now, as becoming an adult, have started to realize that you've given me. And in particular, they were things that I didn't know I needed, but now I do know I need to get through things like a PhD and to have passion for science and passion for challenges. And when people ask me, how did you come to be doing a neuroscience PhD, I realized that my story always starts with you. That, I say my parents are both, do philosophy and have their philosophy PhDs. And I was born when my parents were still in grad school, so I was literally born into academia. And I think just growing up and watching you have passion for challenge, and you kind of instilled critical thinking and logic and the ability to think for yourself and to challenge things, just became a part of how we grew up. And all the times that I went to talks when I was a little kid, I remember being 8 years old and sitting in one of my dad's lectures about infinity, when he talked about like if you have a long line of sheep and you have a long line of farmers and you pair them all up, then even if they're a little bit off at the beginning, they'll still be like equally paired because there's infinite numbers of both. And I was just like, what? But I think those moments just made academia feel like home. And my dad, even throughout forever, has sent me things like, want to hear great paradox? But then send me a screenshot about this. No, Dad, I need to finish my dissertation. Like, I can't look at this right now. Like, okay, well, after we'll talk about it. And so I just want to thank you for all of the ways you've shown me to be excited and to just go after problems. And you have this intrinsic interest that I just really admire. And I would like to thank my mom. This is a excerpt that I found last night and just bawled when I read it. This is from a book that she wrote about pain and then dedicated it to her children. But she even references it, so she addresses the issue. But you have been my biggest cheerleader. And I knew I could always call you when I just had freakouts about things big and small. And you showed me how to just get through it, that you can, that I can do anything and that there's always a solution. And I can't thank you enough for everything. And with that, time for my closed door session.